the AGVision Committee of Zamorano. I am glad to introduce our guest speaker for today, Dr. Timothy LaSalle. Dr. LaSalle is an international researcher, speaker, and promoter of the regenerative agriculture. In this way, he tries to address climate, hunger, water, and farmers' profits. He is currently co-founder and adjunct professor at the Center of Regenerative Agriculture and Resilient Systems at the California State University in Chico, California. He has served in many leadership roles. He was the first CEO of the Rodale Institute, and he was also executive director of the Alan Savory Center for Holistic Management. This is an international nonprofit whose mission is to restore and regenerate deteriorating landscapes. For the Howard Buffett Foundation in Africa, he has been a consultant, advisor, and research coordinator on soil and food security for smallholder farmers. He then worked towards biological restoration of soils for resolving the issues of hunger and climate crisis. As CEO of the California Agricultural Leadership Program, LaSalle worked in more than 80 countries with heads of states, ministers, and community leaders. There, he became a student of many of humanity's global challenges. This exposure to global ecological decline brought dreams and internal tension. It also brought questions about the human impulse to destroy what sustains life. In partial pursuit of this passion, LaSalle turned to depth psychology. His PhD dissertation is titled Awakening to Ecocide. Tim LaSalle is currently Professor Emeritus of California Polytechnic State University at San Luis Obispo, where he also started and operated his own dairy farm. So we will now listen to Dr. LaSalle's speech. Before Dr. LaSalle start his presentation, I would like to tell you that if you have any questions, then you can type them in the chat and then we will discuss them at the end of the presentation. Welcome, Dr. LaSalle.
Well, thank you very much. Muchas gracias. Mi español es muy malo, so I'm going to stay very much in English, and I appreciate your capacities to engage me that way. And I apologize for my deficiency in the Spanish language. But let me share a screen and begin this conversation, if I may, with you today on kind of why regenerative agriculture, and it's beyond a conversation of sustainability. And I want to frame it from this perspective. As I traveled the world, and I've been in over 100 countries, I've been to Zamorano a couple of times in that traversing the planet and looking at agriculture and development and hunger, but also observing the ecological decline and how our 10,000 years of agriculture has not really created much of a bright future because of the degradation that's been occurring. And it pushed me out of my conventional training. It pushed me out of my thinking about high input systems. And it challenged me to think in terms of and find where in fact we could rebuild soil and rebuild systems and pull the carbon dioxide out of the atmosphere that now threatens all of life on this planet. And so the kinds of numbers and the kinds of ways we approach agriculture today that I'll present to you are somewhat revolutionary. They're changing paradigms, but it's where our research is focused now and our data is coming forward in very positive methods or very positive results. Let me just say the way we're defining regenerative agriculture, it describes farming and grazing practices that, among other benefits, can reverse climate change by rebuilding soil organic matter, soil carbon, and restoring degraded soil biodiversity, resulting in a drawdown, not just a neutral, but a drawdown, and improving the water cycle. You know, this is the part that I think that we can't just look at climate change. We have to look at climate change. But really, FAO told us in 2015, we had 60 years of topsoil left. And there's little evidence that would go against this evaluation. As a matter of fact, you know, the soil erosion, basically, David Montgomery, a colleague and a friend, wrote the book, Dirt, The Erosion of Civilization. And you know, things haven't changed. In the upper left-hand corner of this screen was a field right next door to my fields in Africa that was only disked one time, and one rain event took this much soil away. Whereas my fields were no-till, I lost no soils. And as you could see, it is a sandy scenario, but the no-till, and I'll tell you why, actually held it in place. The one in the upper right, my wife and I were doing some work in Burundi when we were on the continent, and every square inch of that country is tilled. And they're sending all their soil down the Nile to the Aswan Dam, losing their primary resource. The picture in the bottom right-hand corner is my neighbor, and it happens right here. It's not just the developing world that's destroying and eroding soils. We do a terrible job with all of our education in how we let our soils disappear. You know, we think we've learned some things since the Dust Bowl in the United States created our soil conservation services and our whole departments in the United States Department of Agriculture, but obviously we haven't. We continue to destroy that which sustains and feeds the world, plus captures carbon. You know, a good friend of mine, Roland Bunch, had published in Africa's Soil Fertility Crisis in a World State of the World report in 2011 that a tragedy was coming to Africa in a famine. And it was going to be a famine, you know, caused by lack of ability to produce food. And it wasn't that he could tell that a drought was coming, that there wouldn't be rain that much in advance. But what he could tell is the soils were having all of their carbon, all of their organic matter farmed out of them. And that means you could have a huge rain event, but you could have famine because the soil's going to dry out fast. It will not hold it. And that did occur. So what it tells us is that we have to rethink this whole idea of tillage that we've been doing for 10,000 years. And many of you are very familiar with no-till environments and no-till systems. I was working with agronomist Kofi Boa in Ghana. And Kofi had gotten his master's degree at Nebraska, and he and I were working on sort of more of a biological system in a no-till environment. And in one of his test plots here, you see cowpeas in the back plot with a good crop on it, and the cowpea plot in the front with a disaster, no real food to be harvested. And the only difference in treatment in his very first year of this multi-year study was tillage versus no tillage. And the no tillage where a crop survived was the plot in the back, and the tilled conventional farming system was where disaster had occurred. And that's just to show us there's one thing about tillage is it can help absolutely destroy the structure of soil and destroy its capability to percolate and then eventually hold water. So these are my soil samples. On the left side is my soil here at home in California. The middle water piece is the soil that's been aggregated. It's crumbly soil. It has earthworm holes in it, but it also has biological glues that holds it together so it won't erode. I forgot that I was teaching a class on my home farm to a group of 80 people, and I gave them this demonstration, and the soil clod would have been on the left side, of course, dissolved from my neighbor's farm where he tills. It just fell apart. Mine held together, but I forgot to take a picture of it. And I left, closed my barn. I went off, I was traveling. I came back 10 days later, and there my soil was still aggregated, still holding together. On the right side, in the center again, is a picture of the soil from the fields I was farming in Africa, that sandy soil. Look at that soil aggregation, holding it together. The water's clear, it's not muddy. Whereas that field that you saw that had eroded right next to it, the same soil, different approaches, it had completely dissolved, and that washes away or it blows away. And so building soil aggregation through no-till and biology is crucial to creating a future, regenerating, building back systems. One of the things that I think is not understood about soil aggregation very much is we know that the weather and the climate is changing, and actually civilization is threatened. Well, in essence, often they talk about flood events and these huge storms that happen and the flooding that occurs as if our soil structure has nothing to do with those flooding events. Gabe Brown in North Dakota, who's very much of the most sort of the premier regenerative farmer, when he took over the farm, he could percolate about 1/4 of an inch of water per hour in his soils. So in heavy rain events, he got pools and flooding and runoff. Now with his biological no-till, he gets 11 inches of percolation per hour. It goes straight down. So think in terms of heavy rain events, how much flooding would be mitigated by soils with good structures absorbing the water and holding it for later use. So two critical principles of regenerative agriculture are no-till or minimum tillage and cover cropping to give a buffer. If not cover cropping, then mulching. Cover the soil, keep it covered, cool it, hold the moisture, and stop the erosion. And also feed the biology. Biology. Here's the new paradigm, even though we talk about living soils, but most of our training has been in the geochemical understandings of soil science. The soil science class I took in the university was the same class my father took 30 years before. It's the same people following 30 years later took, and it had to do with inputs, with geochemical relationships in the soil. But when we move to a living biological soil, a biome, a living process, the whole paradigm changes. And this is the new soil science that's really breaking through our understandings. When the fungal to bacterial ratios in that living healthy biome gets to be at least 1-to-1, preferably 4-to-1, the whole functionality of soil changes from input needs to creating its own nutrient supply chain for the plants. Quorum sensing organisms come together and a self-fertilization normally can take place. The soil carbon capture can move from what Ratan Lal says is a 1/2 ton to a ton maybe per hectare to 10 tons, a whole magnitude higher. And this, there's a lot of people pushing back against this data, but it looks like it's going to prove out, the more replications we do, to be the case. The other paradigm change is crop productivity changes because there's an improved photosynthesis efficiency. I didn't know that existed. You mean photosynthesis can be more efficient? It can, because you can reduce the amount of carbon being re-released to the atmosphere and hold it in the plant. When we take this David Johnson inoculant that anybody can make in their own backyard, that you at Zamorano can make in your experimental plots and try with your plants at almost no cost, you see big changes. As a matter of fact, he just applied it to this sorghum plants in the desert of New Mexico versus non-application, and he got a crop versus kind of no crop. Some people in Belgium took the Johnson-Su methodology of inoculant in cover crops, and they got double the total biomass in those cover crops just with the biological inoculations. In Australia, in a very dry zone, a farmer took these biological inoculations in his barley crop, and he doubled the yield. His farmers next door were going broke, and he now has 20,000 hectares he owns because he's making money and everybody else is losing money in a rain-short area of Australia. The other phenomena that occurred is where you see his barley rows that have been harvested here is these green grasses that are starting to grow. That's a perennial grass that has not been seen in the area in 40 years, but the biology being applied to the soil is resurrecting old seed and giving the right biome the right biological opportunity for it to regrow. This is not now a weed for him. He actually turned sheep in here, and it's a great resource. So what you see on the right are where I saw in Africa, I pulled some weeds out and I saw my roots that look like this, and I thought they were diseased, but it's actually the healthiest root you can see when you have what one of my farmer friends here in California calls dreadlocks on his roots, versus when you're growing with fertilizers and a dead soil, you have these white clean roots like in the left. Well, what happens is you have a dead soil on the left, you have a living soil on the right, where actually the root hair is pushing sugars and carbohydrates, in other words, carbon, into that soil to feed the organisms. It's a relationship that has developed. It's a biome, a biological system that the plant feeds them and they feed the plant. And the health and resilience of the plant is improved. The carbon is being dumped into the soil. Organic matter is being built, better water retention, and a more biodiverse, healthy system from insect and disease resistance. What I've just learned recently too, I didn't know existed, and maybe you all already knew this, but plant root tips, root hairs actually absorb a lot of these bacteria and digest them, which means that a lot of the nutrients are going to come from these microorganisms. They also have places on the root hairs that it expels the wastes, or actually sometimes the whole cell, the individual cell of a bacteria, that then goes out and restores itself and starts to reproduce and produce more, and the cycle continues. This is called rhizophagy. It's a rhizophagy cycle that's occurring. And what we learned at Rodale Institute was the significance of mycorrhizal fungi, how its ability to improve water holding, nutrient acquisition, and carbon depositing. What can happen when our soils become biologically healthy with a healthy biome is that the root hairs of that plant can be colonized by mycorrhizae, and then the root surface has been increased by up to 50 times. So if the root hairs now have 50 times the reach to get moisture and nutrients, you've increased the efficiency, capacity to feed that plant. Those mycorrhizae also demineralize bound up phosphorus, so you don't need the fertilizer. They move nitrogen that's being demineralized and moved to the plant. You don't need fertilizer. And what happens is, in this sort of succession of soils, you move from 100% or bacterial dominant soils, and you have the perfect environment for weeds. Dr. Ling Ingham teaches us that in the soil food web. Weeds do exceptionally well in bacterial dominant soils. When you get up to a 1-to-1, 2-to-1 ratio, you're in where the agricultural crops perform the best. They need that fungi we just talked about. And our soils are almost bereft of fungi for two reasons. One is tillage, it destroys the communities, or chemicals, fertilizers, fungicides, of course, herbicides, destroys those communities. And if you go then to 100-to-1 to 1,000-to-1, you're probably standing in an old growth forest where, of course, you don't see weeds and you don't need fertilizers, and you have great communication between the plants with those fungal communities. Anybody can build and make this inoculant for that soil and restore the biome. It's a simple process. It's a no-turn composting, aerated composting system, where it's not an anaerobic system. It's not bacterial dominant. It becomes fungal dominant. It does take a year for it to mature, because the metagenomic look at that compost early on, this is at 4 weeks of composting, shows about 740 species of organisms there. 80% of them are represented by just 23 species. But by 60 weeks, you're going to end up with about 80% then has 99 species, a whole diversity that is improved. It's like our human gut biome, when it's diverse and healthy, we're healthy, our immune systems work, our moods are better. It's really all proven scientifically. The same thing happens in the soil. And we've got to restore where we've eliminated so many of these interrelated biological organisms that work with each other to create a functioning soil. It takes diversity above ground to feed it as well. So monoculture cropping you can get away with for a season, as long as in the off season or the second season, you're bringing back that diversity, like in a multi-species cover crop, to refeed and restore and be sure that these organisms that you've re-inoculated into that soil are being fed. On the left side of this photo, Dr. Christine Jones shared this with me, is this is in Canada, where there's a single species triticale crop that's failing because of a rain shortage. But the multi-species cover crop in the same field, in the same rainfall, is vibrant. And you see what happens when the organisms and the fungi are available to hold, store the water, but also supply the water and nutrients to the plants. It's robust and doing exceptionally well. Just inoculation in cotton there in New Mexico, you see, with inoculating with this Johnson Sioux, more than doubled the yield versus the normal nitrogen application. I found in my work in Africa, I was on phosphorus deficient soils, but after the first year, just with no-till and no outside inputs, no compost, no fertilizers, I began to get the same kind of yields that other universities were doing next to me with heavy inputs. And it was because the biology could perform. And I no longer had phosphorus deficiencies, even though all soil tests would tell me I had deficiencies. But the fungi could demineralize and bring the bound up phosphorus to the plant. On a project we're doing with Howard Buffett right now in Arizona, in the first year with the corn, first year of inoculation, we were able to almost match the yield of the 256 pounds of nitrogen with no fertilizer. There was 126 pounds of nitrogen lost to the environment, whereas our biological systems created 107 pounds. We had more profit because we didn't have to pay for that nitrogen. And the farmer who managed it, he worked with me in Africa, and this is some of the corn I was producing there. And he's there on the right in this picture there in Africa, but that's what he was doing in his cornfields in Arizona when he walks out. And he's an old Midwestern farmer used to all the fertilizer inputs. And he had an ear of corn from the conventional fertilized system and the only biological system. And his comment was, why am I buying fertilizer? And that's the comment we want farmers to understand. In working with Howard Buffett, one of the things I keep encouraging him to do is to talk about the brown revolution. The green revolution has failed a couple of times in Africa. It will continue to fail. It cannot succeed. And we could go into that if there's questions. But in essence, the brown revolution will be the real green revolution, build the soil. And let's just remind ourselves, time is up on climate change. We have to take that carbon out of the atmosphere and put it back in the soil. This biological system looks like it can capture a magnitude higher, even if it's only 5 tons per acre. We could draw down those excesses out of the atmosphere, restore soil health, fertility, water cycles, stop flooding in many cases, versus the conventional research that's out there with Rattan Lal or Urs Niggli in Switzerland or West. We are seeing biologically how this works so much more effectively. Gabe Brown shows the same curve on carbon capture in his multi-species cover crops with grazing as David Johnson shows with just the inoculation. We know that this has happened on his farm. We know that it's happened in the test trials, and we're seeing it happen in our 5-year studies that we're starting to move forward. And fingers crossed, it shows up with similar data points, but it looks like it just may. What we're saying is this: when you go into this fungal dominant world in the soil, you can increase soil carbon by 10 times. and you can reduce soil carbon respiration by 4 times. That means drawdown. That means regenerating the health of the soil. And that means building a system for the future. We know that in planned grazing, amp grazing, we can take the desert in Sonora, Mexico, like this, and turn it into a grasslands that it used to be before we mismanaged and abused the grazing, as we historically have done on all continents of this planet. That captures carbon. That's a living system. It photosynthesizes every day versus a bare desert soil. So we have a chance through regenerating our biological systems through a regenerative approach of actually drawing us down to pre-industrial levels and building a future for our youth to be able to feed the world and restore the biodiversity. With that, I will just say that David Johnson is now an adjunct professor with us. I'm an adjunct professor up there at Chico. Cindy Daly's our director. I think she's going to speak with you all later in the year. We are very, very proud and excited about the results we're seeing. We'd love to share them. We'd love to see other people do it themselves, do the research. Let's learn together. It's too important to ignore it. Please let me respond to questions or comments that you may have.
Video. Thank you, Dr. LaSalle.
Thank you.
It was an interesting presentation. We have questions in the chat, and please, everyone, if you have any question, please send us the question through the chat. The question that we have is, what is the essence of making the Johnson inoculant and how to use it?
So if you go onto our website at the Center for Regenerative Agriculture at Chico State University, there is a landing page with Johnson Sioux Bioreactor to register it, and there's films on how to make it. There's PDFs on how to build one. I'm actually building smaller ones at home. I was just at the Piscinas Ranch yesterday. They built a huge one. And so we just need to hold to some principles, and then we need to test it at the end to see if we've succeeded to get the fungal dominance correct. And it's a natural process. I used to call David Johnson and say, well, what's in it? Well, what's making it work? Well, what am I looking for? And he's reminded me, everything is in it. He says, I've even found microorganisms that we thought were only living in Antarctica, but he said, they're even in my bioreactor. And the point is that when you put in a lot of brown material, and that means leaf matter or grass or hay, and you have it chopped up, and it can be cow manure, etc., not greens, not like you do a regular compost pile, and keep it aerated. It won't heat up. It will not go to the thermophilic stage. It'll heat a little bit, but it cools down. It doesn't get as hot. You put redworms in and you just keep it wet. That will collect everything in the atmosphere, and all the spores from the fungal communities, they'll all show up eventually. And so in essence, you end up with tremendous product. We've had people send the metagenomic analysis from Australia with different feedstocks, and even eucalyptus, which I thought nothing can grow in that. Eventually, after this 12 months, with the worm activity, with the aerobic conditions, with the inoculation that comes from the air, you end up with a very healthy biome, but it has to be monitored and managed well. There's some big research project going on in Kenya right now. They paid attention. My wife and I visited them in 2019. They built a beautiful bioreactor. We know that every continent except Antarctica has these bioreactors going now. 61 countries are following this and engaging it, and thousands of people. So we all need to do our citizen science, or in your case, hard science, with inoculating the seed. And we find that most efficacious, which simply means you can take a small amount out, stir it up in water, shake the organisms loose, put it through a paint filter or some filter, and put it in a spray unit and spray the seed. We're building on farm scales here, drill seeders and row planting seeders with like what would be a fertilizer injector But of course, we're not putting fertilizer in. We don't want that in. We're putting the inoculant in. And as the seed drops, we're spraying the seed and pressing the soil on top. That way, the organisms are there, like in the birth canal. As humans, we get our biome inoculation when we come out of the birth canal, and we meet the world with the organisms that help keep our skin healthy, that help keep our gut healthy, and then we get them picked up from healthy soil, from skin surfaces of our family, from the dogs that run through the house. That makes us healthier. Even though there's pathogens in there, it's all there. It's in the right proportions, and it gives us immunity, and it gives us a response to the world. The same in soil. It's all in there. It's just that with the fungal dominant, it's the healthy system that's going to work effectively. And so when the seed is inoculated and that root hair comes out, they start their association immediately, and the plant begins to work. It's not fed an artificial sort of injection of the nutrients where it'll shut down and doesn't have to feed. It has to feed that organism, and the organism has to feed that plant. And so the healthy relationship begins at the moment of the seed emergence. And that's the beauty of it.
And in relationship to what you were talking right now, do mycorrhizal fungi enhance soil sequestration and soil health?
Seems to at a very high level, but part of the thing that I think is amazing, and I don't like the word sequestration, partly because it's sort of like captured carbon that'll stay there forever. Well, that's sort of like the biochar, the terra prietas, or that sort of thinking. And that can be dead carbon. We actually want living carbon that's going to build life in the soil. What we just want is more of it collected and held on a longer time frame. Just keep collecting, respire some, but collect more and more and more, but cycle it through the living system of the soil. So I call it carbon capture, or I call it carbon accretion, and that's what we're looking for. Mycorrhizae, the glomalin that it secretes to glue soil particles together to help make an aggregated soil, that we know lasts for about 40 some years in the soil. Unless you're tilling it and oxidizing it, it'll last a long time. There are other things when that soil particles are being glued together. Sometimes they're gluing and capturing carbon and holding it in those spaces from organic matter. But we know that over 50% of the soil carbon could come from the microorganisms themselves. So as you're feeding and growing the mycorrhizae that will extend, the root hair surface 50 times, that's that much more carbon down below soil line than above in the atmosphere. So that's capturing and building beyond leaf matter and decaying plant matter. It's growing the biology. It's going to increase and feed earthworms. That's carbon. They are carbon. And here's another thing that's often not looked at by soil scientists, and that is what we're also capturing is in the oxalate cycles of these organisms, is we're they are depositing inorganic carbon in the form of calcium carbonate. So you're accruing an inorganic carbon, again, coming out of the atmosphere through the biology, deposited into the soil. So all of this builds it at a faster rate. And why we think, as much as we're attacked for our numbers, we think they're going to keep proving out, is that when you're dealing with a functional living soil, it's different than all the measurements being done on a broken semi-dead soil that has been either repressed in its fungal communities, particularly by fertilizers or tillage, that represses and kills that, to one that's robust in its particular living systems. I can read others if there's, what is the difference between the use of only conservation practices for soil and the use of regenerative? Well, conservation, a lot of conservation agriculture, and I've worked in that field, when it's done in a high-tech GMO Roundup ready fertilizer-based system, you're going to get the kinds of numbers of carbon capture that Ratan Lal reports, which is minimal. and you're not going to you're going to capture a little carbon to a point and then it stops. So you're not going to really rebuild systems or capture large quantities, and you're going to be stuck in paying for the cost of all those inputs. And that makes it harder for farmers to profit. So from smallholders to large holders, if they can avoid writing those checks or paying for these inputs, they can make more profit, and they're not going to diminish yields. I mean, that's not our experience. And in some cases, like with David Johnson's work, yields continue to increase year after year as that soil gets healthier and healthier. So without those outside input costs and continued growth in yields, the farmer does better and better.
Do you want to read, because we have questions before. So one of the questions was, what would be the difference in applying regenerative agriculture in a tropical region versus a temperate region, mainly talking about soil resilience?
So that's the question we want Zamorano to answer for us. I don't know, but I'll tell you, it is, we have some bioreactors going on right now in Costa Rica, and we have some, of course, in Africa in tropical areas, and we would love to know if there's a difference. We would love to know the responses and results that you get from those trials. And so it has to be done. And we think there's no reason it's not going to work there too. As a matter of fact, it might be more robust because you're going to have longer growing seasons. You're just going to have to do some rotate or interspecies plantings so that you're not just single monoculture crop after single monoculture crop. Remember, diversity below ground is reflected by diversity above ground. Diversity above ground is reflected by diversity below ground. So you have to bring complexity back to the system. It doesn't mean you can't grow a corn crop. You can go grow maize, that's fine. But when that crop's done, come back in with something more complex. Do a cover crop or interseed your maize later with multi-species cover crops to feed all that other biology that you want to maintain. So when you rotate out of corn to the next crop, you have the organisms present for this self-organizing system to adjust to what that plant needs for that growing season. When you single monoculture crop and just keep doing that, you start to limit. It's like if you only ate tortillas, or me, if I only eat ice cream, or whatever you like, you might just only like steak, and that's all you eat, your gut biome will change. You will not be healthy. My wife makes me stop eating only ice cream, but that's what I'd like to do. But I wouldn't survive very long. The biome would change and I'd be dead. Same with our soils. So we need the rotation, the diversity, just like for us, for the nutrients, for this case, the different organisms to function.
We'll take on your offer. I'm doing some research down here.
Wonderful.
Yes. So in terms of regeneration of soil, specifically in the biological part, what is your comment as comparing using or incorporating crop residues versus cover crops?
So incorporation is again going to disturb, and we're going to go, no, please, don't. And that's going to disrupt the fungal communities. So, all of that residue from the crops left on the ground, you know how fast, particularly in tropical environments, how fast that can decompose. That is feeding life. It's feeding the organisms. And it will help stop flooding. It will help hold moisture, and it will feed organisms. So leaving the residue on top is really a crucial component. So if you're not inoculating and getting this biology in right away, and you're just starting with no-till and residues and that, the transition to a healthier soil is slower. But if you combine it with the inoculant, you'll see the boost immediately, and then you're in a maintenance and feeding mode, and it'll just keep getting better. So the whole thing is, like David Johnson would say, is if you're just doing your multi-species cover crops and rotating that with your cash crop, you'll eventually get to the levels he gets to, but it will take you 20 to 30 years longer. It's just a slower process. You are rebuilding and reestablishing the organisms, but it's longer than if you just inoculate it and put the tens of millions per square foot in that field of these organisms. You've just restocked it with the below ground, what we call livestock. It's not four-legged livestock, it's singular cell livestock, the living system. You've just reestablished it very fast. And then that gives it that kickstart boost, which will help the system go.
A while ago, you mentioned biochar. Do you think it could be used as a supplementary practice to increase fungal activity in the soil?
I think biochar will help definitely on your carbon numbers in soil. Apparently there are plant responses. I'm in conversations with people on biochar all the time. What I don't know, and I'm not a biochar expert, I know I'm gonna be learning more and more about it because we're probably gonna do a project where we will inoculate it with Johnson Sioux compost and put it in the soil as well. But I always heard a conversation around biochar as saying, well, the biochar is like a hotel for these organisms. It houses these living organisms. But what I don't understand is that the rhizosphere of the root is where all these organisms live, because that's, I mean, not all of them, but where the highest concentration is, because the plant is feeding them and growing them for its own health. So that's where the concentration is. Why would the plant leave its feeding center, its sugar and carbohydrate center, and go check into a hotel so where there's no food? Because that carbon in the biochar is kind of a dead carbon. It will hardly degrade at all. So I'm not understanding it well enough yet. Would I use it if it was available? Yes. Would I go out and burn my corn stalks to make biochar? Absolutely not. That corn stalk should be used by the biology of the soil in that carbon cycle. If it is, in our case in California, where we have a lot of walnut shells and pistachio shells and maybe almond shells, we can biochar that because it takes a long time to break down and it's not really that accessible to the organisms of the soil. So we could make that into biochar. We're going to start to do some studies, it looks like, where we'll start to evaluate it. But it's not directly part of this living system. Might it support it? Maybe. Am I telling you, yes, go do it? I don't know. I don't know. But please don't destroy the soil food web's source of carbon by burning it into a hard, dead carbon. We want the living carbon for those organisms.
According to your experience, is it easier to work on regenerative agriculture with small and medium scale farming than large scale farming?
No, I don't think it's easier or harder either way. It's just the principles are, it might be has to do with the equipment or the tools or what you're going to use as to how difficult it's going to be. It's a matter of attitude, of managing the system, paying attention to the system, and knowing regenerative is a little more complex. It's not just a cookbook, plant at this date, spray at this date, add fertilizer at this date. It's not like that. It's about building the system and working with it, understanding nature and supporting her in a very robust way, and paying attention and figuring it out.
So in Honduras, as many other countries, we have promoted practices such as crop rotation, no tillage or minimum tillage, agroforestry, and we continue trying to impulse practices such as agroecology. What would be your point of view on what is the problem on the adaptation of these kinds of practices in our growers here in our countries?
One of the problems for all of us, you as scientists and teachers and professors and farmers, me as a farmer and a former professor and a scientist, has been the biggest challenge for us is our old training, the way we were educated, the way we practiced agriculture. That's a barrier because we have a mindset of input. The plant needs nitrogen, bring it nitrogen. Not understanding the atmosphere is 78% nitrogen. It's all right there. Do we have the air spaces for it to get down there? Can it be incorporated in the biology, in the plant? Yes, it can. And so the whole thing about my fields in Africa that were phosphorus short, every soil test said these soils are phosphorus short. No, they weren't. The mycorrhizal fungi demineralized, bound up phosphorus that they said was not available to the plant, but it was biologically available to the plant when you worked with the organisms. So that's our biggest constraint to being able to make this change. It's our old training of a geochemical model of soil instead of a living biological creative soil that can respond to most all needs of the plant, and they work together. When you build that, your bricks level and health of the plant gets better and better. We've seen examples of that. When that happens, you end up with less insect damage. So there's a study that was done here in California with almonds. 4 conventionally farmed almond orchards with 4 regenerative almond orchards. One of the regenerative almond orchards not only had cover crops, but it had sheep grazing in there in the non-harvest season. So it even had livestock involved in it. What they found was the same amount of insect damage from the conventional almond orchards that sprayed for the orange worm that damaged almonds, the same amount of damage in the conventional system that did 0 spraying. So the regenerative systems had the complexity, had the predators, and had probably the bricks levels of the plant itself high enough that the insects left that orchard and went to the conventional field, where through chemical agriculture they had been stressed, and those are susceptible, more susceptible to insect damage. The farm in Arizona, where the farmer, the manager there, who was in one of those pictures, said, he said, I know you don't like sprays, Tim. I said, listen, we just need to figure this out. Let's try what we can do. He said, well, I'm going to have to spray twice for mite in this corn. We haven't talked about it, but I know for a fact he only ended up spraying once. And this is going to be our 2nd time with corn. This is our 3rd year. And I'm putting money on the table. He won't have to spray it all this year. That it just will be where the soil is healthier, the plant is healthier. Those mite are going to go to another field where the plants are weaker and tastier to them. So that's, Chubosco had done studies on that in France years ago in vineyards. Lundgren's doing studies around that from his Agdices Foundation. It's being shown that the healthier the plant, insects disappear. Also with the biodiversity, for every predatory or damaging insects, there's like 700 to 800 or 1,000 predatory insects that will keep them in check. So we start to save costs for farmers when we regenerate a healthy biological system and a more resilient plants for them.
Okay, so if you had the opportunity to talk to one of our growers here in Latin America, for example, what would you say to these farmers in order to convince them that this is a needed change?
So one is that, I came from the conventional world, went to the organic world, and that's just a fight between organics and conventional. Oh, you don't get enough production in organic, or you guys are, purist, or we can't feed the world. And it became a contentious fight. And I think we have to be careful with language and not create a language or regenerative that sort of says it's an ideology or a philosophy, or this is the only way you have to do it. I think it's a matter of supporting farmers to make stepwise transitions to things that build their system where they're seeing they have less cost and they're getting the same or better results. And so it may be this step, then that step. That's the beauty of this inoculant, is that typically you can see the results immediately. And then the farmer, just like Doug Aller in Arizona, said, why am I buying fertilizer when he doesn't have to buy fertilizer? He actually he's made a whole bunch of his own bioreactors on that farm. And with Howard Buffett, unbeknownst to me, Howard just told me this recently, they're doing trials now in Nebraska and Illinois all on their own because they saw the results in Arizona. And that's the kind of thing we'd like to see happen, is, have farmers try this. Pretty soon other farmers will try it. They'll all learn and see how it works in their systems and figure out what's not, why, if it's not working, why, and what needs to happen. So it just, when we look at a healthy, functional, living soil, it's self-organizing, it adjusts. The quorum sensing can actually help create what the plant's signaling, because the plant signals what it needs and the organisms respond. It's just like there's a good book out now called Finding the Mother Tree about forests and how the mother tree, through fungal communities, will send carbon and feed plants that can't even, that live in the shade and can't get photosynthesis, but it will send the carbon so the baby plant can keep growing. The same interrelationships can happen in agricultural fields. We're just learning about this. But what we can do while we're learning is we can begin to implement and study it and watch the response and results. This is a new paradigm. It is a new world for us.
You work in different countries. Do you know if there's any countries where they have any policies that try to improve or try to differentiate the products that are grown using sustainability or regenerative agricultural practices?
Policies that are reviewing them, is that what you were saying?
So policies that try to provide incentives on.
Oh, yeah. There are actually a few policies in the US that look like they may come forward. Not good enough. I'm sitting on a committee right now with 2 former US senators that are trying to get some policies in place. Australia is doing a little bit more than the rest of us in the world on this. And their understanding with professors like Dr. Christine Jones and a couple of others there, that they're understanding this inoculant piece a little faster than some other parts of the world, and the fungal communities and what they call the liquid carbon pathways. So I think they're a little ahead of the world. Europe is lagging. US is lagging too, but we now have, there's another regenerative ag center that's just been created at University of Missouri, and we're going to be in conversations with him. There's a regenerative ag center trying to get started at Clemson University in South Carolina. We've been in communications with them. There is a university, Southern Cross University in Australia, that started a undergraduate program in regenerative agriculture with really good professors coming to the table there. We've been in conversations with them. And then there's, as I said, the research that's now going on globally, and we're in more and more conversations with those people too. So it's a matter, I think, of supporting, asking these questions, trying to collaborate on investigations, trying to learn what nature wants to teach us, how we support her, and help the system become regenerative and robust. You know, those dark, deep soils in the Midwest of the United States were not made without animals grazing in deep-rooted grasses with fungal communities attached to those grass roots. Those 3 pieces, the biology of the rumen, the biology of the saliva of the animal biting the grass, the stimulation of the biting of the grass to cause more of that root exudate, the sugars and carbohydrates, to feed the organisms and the fungi so it can regrow again, captured carbon and built deep black soils. That's all we're trying to imitate or support right now, is that same idea of what nature figured out and nature and how nature works.
So we're running out of time, but we have a lot of students here in our meeting. So what would you tell them as future professionals in agronomy? Like, what would be your final thoughts on what they should be looking at the future?
As students now, and the age that you are, climate crisis is not something out in the future, it's present. Your actual livability on this planet determines of whether we can draw down enough carbon to bring us back to in a living, more stable climate so food can be produced, so storms don't destroy so much of our land and of our communities and civilization and disrupt the economies. And so finding more robust ways to do that drawdown, it looks like improving photosynthesis efficiency, capturing more in the soil is the cheapest, fastest, quickest way that has a whole bunch of other ecosystem services on water retention, on cleaning up waterways, of getting nitrate pollution and phosphorus pollution out of the oceans where we're killing so many, creating so many dead zones. Regenerating these soils is the only way to build a future. We have overshot the chance to be sustainable because we are over consuming and over polluting. We have to restore, rebuild, and regenerate. And so I just say to all you young students out there that are now in the midst of this and with your professors and teachers here, is have an inquisitive and curious mind. Don't take anything I said as the absolute truth. Question, investigate, and give it a run, because we have to get creative and innovative and work with how nature teaches us. It knew how to pull that carbon out of the atmosphere when it was over 1,000 parts per million through photosynthesis, through burying it in the soil. And then we dug it back up and burnt it and re-released it. Let's get back with what she knows and recapture it and put it back in that soil. There is a chance for us to do this, but it will take all of us to do it. And so I just invite you in, ask the questions, investigate, be curious, and give it a try.
Thank you so much, Dr. LaSalle. We really enjoyed, it was a really interesting presentation. I think we all learned a lot about regenerative agriculture. Thank you to everyone that was on this meeting today. Just to remind you that we'll have the next meeting on June 24, and we will hope to see you all in the meeting. Thank you again, Dr. Timothy.
Thank you.
All the best. Thank you very much. Bye-bye.
Bye-bye.